All right, hey, good morning, guys. This is just a quick tutorial for how to use a program that we're gonna use for our cloning experiments called SnapGene. SnapGene was great and sent us some free files, some free downloads, so that we could kind of play around with their software a little bit and use it for our experiment. So huge thank you to them. In order to get that download, you're just gonna need the link that they sent me. So where I put that is on Google Classroom. Okay, so when you go right up to the top of Google Classroom, there's going to be this portion right here. That's just the SnapGene link. All right, so if you click on that, it's going to open up a screen for downloads. Right, so you guys are going to be working on the Dells in class here, and what I'm going to want you to go ahead and download is the one for Windows. So you'll download this version here. You guys can see there's a couple of important bits of information. One is the group name and one is the registration code. I don't think you'll need to type these in anywhere, but just for reference, if you need them, they're here. You're also going to need me at some point as you're downloading to override your computer, and if you need that, when you need that, just call me over and I'll type in the stuff that I need to type in, and we'll get everything downloaded and up and running. Okay, so all you really need to do is kind of click on this and follow along. If you need my help, just call me over as we're downloading today. Okay, so you'll hop on a computer and you want to get to this link, and then you'll just want to click on Windows to get the download. Now once everything is downloaded and ready to go, what's going to pop up is SnapGene. Right, so you guys will have this on your desktop, and you'll have a place where you can click up here, right? So let me expand my screen a little bit so you guys can you can see this. No, it's not going to show. Anyways, you click on SnapGene, and when you click on SnapGene, you can open File, and you guys are probably going to be able to see that right there. And there's several different options. There's New DNA File. Maybe if I just have like the A's, the C's, the T's, and the G's for a particular sequence. There's another thing that I can actually do with SnapGene, which is kind of cool, which is just go like search or import. So we're going to try this first with you guys, and then I've got a backup here using Google Classroom if we need to. What we're going to do with our cloning experiments is we're going to cut one gene out of a plasmid, a gene of interest, and then we're going to try to move it over into another plasmid. Right, so what I want you to do is go ahead and click on Add Gene Plasmids right there. Okay, and then what pops up is just like a search tool, just like Google. You can search for terms. You can do this just with Add Gene Plasmid. So it's like Google for Add Gene. And there's two plasmids that we're going to use. Okay, so these two plasmids are PJK. Okay, and then it's underscore ProK1. And then it's EGFP. Okay, so here's the first plasmid that we're going to use. So they have all these kind of silly names, and what we're going to want to do is go ahead and click import. All right, and when you click import, it's going to download that sequence for you. And now you can see there's a plasmid map that pops up. So it's a circular bit of DNA, and it's got all these different elements on it. What SnapGene gives you is really just two main views. One is the map, and the other is sequence, right? So what we're currently on right now is called the map, in which it gives you just these different elements where you can kind of like take a look, right? And you can see what these different elements are that are all in different colors. The other view is called sequence view, in which you actually see the A's, the C's, the T's, and the G's, which is this right here. So I just clicked on sequence, right? And it's got all the same elements here. It's just giving me the actual sequence of everything. And this is going to be really important for we want a little bit closer view of the plasmid, right? So you guys can see, you can just scroll through there. And we've got a view of the plasmid, right? So this is one of the plasmids that we're going to use. Now what we're going to try to do, our attempt is going to take this GFP gene, this green fluorescent protein gene, and we're going to try to cut this thing out. And then we're going to try to, when we cut it out, we're going to try to paste it in another vector. That other vector we also need to download. Before we go and do that, what I'll want you to do is hit save, right? And then we're going to hit save here. And then I'm going to want you to save that in your SnapGene account. And so we'll save this as PJK ProK1 EGFP, and then put underscore and your initials, right? So you'll just put your initials here, underscore, and then the period. So we'll go period 1, just like that, right? So then we'll hit save. And now that will be saved and ready for you to work with. Okay, so there's one that's saved. I'm going to close this one out, and we're just going to start over again. So I'm going to go to SnapGene, and we need to pull up our other file. So I'm going to go to import. I'm going to go to add gene plasmids, and my second plasmid is going to be called a PET, PET 28, and then we're going to say noser, which hopefully this will pop up. Now this one's going to be harder to find. PET 28, it might be B. Noser. Okay, so if this one's not popping up, which it looks like it's not going to for me here, I put a couple of other sequences that you might need, right? So on Google Classroom. So if you hop to here, I put both files located right here on Google Classroom. So here's the one that we just downloaded first, the eGFP plasmid. The second one is PET 28 noser, right? So that's the one that I was trying to search for, but it doesn't seem like I can pull it up. But if you click on this, what we can do is go ahead and just download the sequence, and then if you can click on your download, and up pops the plasmid. So I'll just have you guys download that one directly from Google Classroom, and this is the second plasmid that we're going to have. Now this plasmid is going to be called a vector, and the reason we're using this as our vector is it's going to allow us to overexpress a protein of interest. So remember with the last plasmid, what we wanted to do was use scissors and cut out just the GFP gene. We just want that. And we're going to try to move that GFP gene over here and paste it into this vector right here. So that's going to be our goal. Same deal here. You can look at the map, and then you can also look at the sequence of the A's, the C's, the T's, and the G's and see what's happening. What I'm going to want you guys to, again, to do today is just go on up and hit save. Right? You've already got the name of the plasmid that should be popping up. Maybe. Yeah, we've got the name of the plasmid popping up. We're going to go underscore again, and then we've got your initials, underscore period 1, right? And then we're going to go ahead and save that. Again, we're going to save that to SnapGene, just like that. Okay, so now you've got your plasmid actually saved to this. Okay, now there's 3 main parts where you've got these pulled up that I'm going to ask you guys to go ahead and look at and kind of just explore around a little bit. 3 things that I'd really like you to pay attention to and jot down are, number 1, the antibiotic resistance gene for both plasmids. So can you find the antibiotic resistance genes, right? So on this particular vector, you can see if I'm hovering around, there's little bits of information that are going to pop up, right? And there's 3 things that I'd really like you to pay attention to for now. Here's the first one that I'd like you to pay attention to, right? So there's the antibiotic resistance gene. So this confers resistance to kanamycin in bacteria. So this is going to allow our bacteria to be resistant to kanamycin. We're going to talk about why and why that's a good idea. But I need you to jot that down for this vector, that what we have is kanamycin resistance. The second element that I'd like you to look at is just the origin of replication. Right? So here's our origin of replication for this particular vector. And notice what it says, right? It's 589 base pairs, and it is a high copy number. And then it's got the origin of where this thing comes from for the origin of replication. So there's the second important element on a plasmid. So we've got antibiotic resistance gene is very important on a plasmid. The origin of replication is very important on a plasmid. And then we've got our gene of interest. Okay? So here, we don't necessarily have our gene of interest. Remember, what we want to do is paste in our gene of interest. That's what we're going to try to do. This one is going to be something that's important for us later down the road that I'll talk about. So what you'll want to do is go ahead and open up the other plasmid, right, the eGFP plasmid, and that's going to be our gene of interest, the GFP. But what I also want you to do with that other plasmid is tell me what's the antibiotic resistance gene and what is the origin of replication. And we're going to want to write those down. And then just kind of explore around a little bit through SnapGene, write down any big questions maybe that you have as you kind of look at this data, things that you think maybe are important, jot those down, and then we're just going to have a class discussion. But really what I wanted you to do today, the main goals, are to download SnapGene and get your 2 plasmids that we're going to go ahead and use saved. Explore around a little bit just so you can get familiar with the software, and then we'll really dive into some details tomorrow. Okay? If you guys have any questions, make sure that you write those down so that we can talk about them in class.